Oxford Nanopore platform. So the thing is that what are the basic advantage of that? Now, basically long reads. So now thing is that one of the major advantage is that this is very small in size. This is very small in size. Now you know what? The last day I have shown you the platform for the PackBio platform or Illumina platform. So these are not basically a benchtop platform. So these are very high in size. So you can't basically take this thing out of your lab. So the most advantageous at this moment is basically the Oxford Nanopore platform, where we basically as because the size is very small, we call it a pocket sequencer, just a bit bigger size than the normal USB device that we use, like a USB device that we can use. So we can use the platform like this. So there is one USB port. So same in case of your Oxford Nanopore platform. So this is a very computer friendly also. So whatever the data that you have captured, so you can take the data. During the data capturing, we can put the system in the PC, and we can curate the data in our own system. Now the second one. So first one is the size is very low, very good. So the advantage is that wherever you go for your data collection, maybe you can go in the forest. So maybe you are a scientist on a background of so you are a scientist that uh, you deal with plants. So you have to roam around the different forests. So you have to collect the sample. You have to go for a data because maybe you are far away from your lab. So you can't basically afford to take the sample, then you come back to your lab, then again sequence it, again go back to your place where you basically started collecting the sample. Maybe if you're a marine scientist, so you have to go for a sailing for a longer period of time. So either these are the cases where you can just collect the sample. You just use any small kit where you can you extract the DNA, and you can go for your sequencing. As this is a server based, so whatever the data that you have captured, you can go and for a quality checking through the server edition. So that is the advantage of that. So I think so some of you still have in its unmute condition. So I think this is creating a lot of background noise. So put everything the microphone in the mute mode. Okay, fine, very good. So the second thing, so first thing is that the long read. Second one is the it's a pocket size, it's called a portable. Third one is basically it's a real time sequencing because you are taking you have isolated the data, you have isolated the genomic DNA or whatever. You can go for RNA sequencing also, and RNA sequencing would be much more feasible because you don't need to break down the double strand. So as the RNA is single stranded, it can easily incorporate through the pore. That is we call it a nanopore. So I'm going to discuss in the next phase of the class. And affordable for individual labs. Why affordable? Because it's very interesting to know that uh, one basic one basic Oxford nanopore sample with 50 sample preparation or 50 sample run, it costs us about only 1.5 lakh only. It costs about only 1.5 lakh. So if you can afford 1.5 lakh, 1.5 lakh taka only, so you can go for a sample sequencing up to 50 sample, okay. Now the point is that in between the sample, you have to make sure that you are following the cleaning procedure meticulously, because this is the recommended thing, this is the recommended part, that in between two sample you have to rigorously clean your nanopores, you enter the channel through which your sample will move through the sample vial to the nanopore. So once you buy this sample kit, so they will again provide you the cleaning kit also, so you can clean the sample also. So this is basically the chemistry. This is basically the chemistry of the nanopore. Okay, this is basically the chemistry of the nanopore. So here we have a cassette. You have a cassette. So in the cassette you have this a nanopore kind of thing. So you have a nanopore kind of thing. So this is called a nanopore kind of thing. So here you have one enzyme. So what happens? This enzyme basically denatured the DNA. Denature means it basically separates the two DNA strands. So once DNA strands basically move towards the nanopore, and once the single strand is basically go inside the nanopore, it basically gives you the sequence. It basically gives you the sequence. Maybe it's a what is basically passing through the nanopore, whether it's a G or it's C or it's C, it's again G or T. So like this. So how this basically is done. So before that, I can tell you one thing. Okay, so fine. Why this is not moving? Okay.
Sir,
So yes, yes. Tell me.
sir, what happens if we take an RNA sample?
So that's why I
In
said
that case,
the RNA sample is very much feasible for this system, because in that case you don't need to use that enzyme, so that it can basically denature. So
okay,
RNA sample is very easy to work with that, because many of the people they are right now using this nanopore technology for the sequence of the RNA, because as the DNA as is a single standard, so it basically minimizes that step. Okay, so denaturation part. So this is the entire thing. Okay, so now okay, so I will go back to the earlier slides. So just see, this is the entire setup. We call it a Oxford nanopore system. This is the entire setup. Okay, so basically the software that basically used for the data acquisition and data curation is basically called albacore. What is the name?
albacore.
Albacore. So albacore is basically the software, or uh, the data curation platform that has been used for the uh, because you know what because you know what this system, once you load your sample, maybe a DNA or RNA, it basically gives you the sequence in terms of the nucleotide peak, like A, T, G, C, whatever. But thing is that this albacore, once you basically call for the data, we call it a data calling or base calling. So once you get the data, suppose you have got like A, T, G, C, C, A, G, G, whatever. But once your software says, or your once you attach this system to the PC, your PC means it's not basically one PC. It should be at least one workstation type, so that during the data acquisition, so your PC should not get in any sort of trouble. Maybe so that's why the workstation, it has basically a high RAM capacity. Okay, so that the data can be processed very fast. Now thing is that whatever the data that you have basically acquired or captured, so there is the second step, we call it a base calling. The second step is basically called the base calling. Now the question is that what is base calling? So base calling is nothing but whatever the base is in terms of the sequence that you have got, maybe A, T, G, C, C, A, whatever. So there is a dedicated statistics behind the data curation, that the base calling will give you the accurate idea that if this is a G, during the real time acquisition of the data, if this is the G, then what is the probability of the G that should come, whether it is a 25%, whether it is a 40%, whether it is more than 60%, or whether it is more than 75%. So thing is that as you can also achieve a paired end sequencing, as you can also achieve a paired end sequence, that means you have a double stranded DNA. So double stranded DNA is basically separated. Now the one sequencing will go from this side to this side, another sequencing you can achieve from this side to this side. So as this is a paired end sequencing, so what happens, whatever the G or whatever the C or whatever the A is basically captured, so base calling will give you an accurate idea that what is the percentage of that G or C or T or A, whatever. So there is one statistics that if any base calling is less than 70%, if any base calling is basically 70%, so that system does not basically that the consider that base as a appropriate nucleotide. So that's why for a initial troubleshooting, you have to go for at least 2 to 3 runs for each and every sample, and that is basically required. Now you can't basically conclude a sequence if you perform the sequencing in any platform simple just once. So for that you have to at least perform a triplicate just to conclude that whatever the data that you have got, that is absolutely perfect. So again here also, so at least 3 to 4 times you have to run the sample to get an exact accurate data. So now if you just focus on the image, that you will see that uh, uh, so there is one non-consumable part, so there is one non-consumable part. So this is basically the non-consumable part. So you have to purchase this thing once in your lab. So this what I have basically circled with, or have a circle with this lower part. So this is called your the non-consumable part. So non-consumable part is nothing but a cassette. Non-consumable part is nothing but the cassette where you basically put your this flow cell. So this one, the upper part. So this one, the upper part. So this one. So this one basically called the flow cell. This one is basically called the flow cell. So flow cell, in the flow cell, there is one small groove where we put our sample, and you put the flow cell in the cassette, and you close the cassette. And you know what? There is one USB device. There is one USB port. Can you see that here? Small one. Can you see this?
Yes,
Yes, sir.
Rajesh.
Okay, so now just you have the flow cell, the top part, you add your sample, you set the flow cell in the cassette, you close the cassette, and you attach the entire cassette, including the flow cell, with your workstation using
স্যার
one.
উপরে ওটা কোন পার্ট বললেন?
ওপরেটা হচ্ছে কনজিউমেবল পার্ট, মানে যেটাকে তোমাকে চেঞ্জ করতে হবে মাঝে মধ্যে, বিকজ that
ও
this consumable flow cell has a
হ্যাঁ
shelf life. This consumable part
কেন
has
অসুবিধা
a
হবে?
কি কি বলছো?
ক্যা ছি ছি
মানে what does it mean?
ছি ছি
এইসব বন্ধ করো, this is a class, বন্ধ
তো
করো।
অভিক্ষিত বন্ধ কর।
Anyway, so
স্যার
yes tell me.
বলছিলাম যে এই আপনার এটা 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 যদি আপনার এত চিপ হয়, অ্যাকিউরেট হয়, তাহলে এটা অন্য অন্য মেশিনগুলোকে রিপ্লেস করে দিচ্ছে না কেন?
No,
বেশিরভাগ ল্যাব তো
স্যার
no,
এটা কতটা অ্যাকুরেট?
আমি বলছি, আমি এই পার্ট
যেমন
আমি
প্যাকটাও যেটা ওটা
আমি আস্তে আস্তে আসছি এখানে, আমি আস্তে আস্তে আসছি। So basically thing is that this, as I said, that this flow cell has a shelf life. Okay,
ও
so initially once you purchase this combination, so it cost around 1.5 lakh. Okay, so now that will give you a maximum sample run of 50 sample run. 50 sample run, and this flow cell, as I said, as this is based on the nanopore technology, so the flow cell has several nanopores on its surface. The flow cell has several nanopores on its surface. So the thing is that sometimes what happens, due to inadequate cleaning, or we call it the cleaning in purpose. So if the CIP of a particular instrument like this, if it is not done adequately, so sometimes what happens, suppose initially you had 1000, so normally there are 960 nanopores are found in the on the surface of the flow cell. Okay, once you basically purchase this. Okay, so basically 960 flow cells are basically found. But sometimes
হুম,
what happens due for the inadequate cleaning, or maybe as this is a kind of the system which is a kind of automated, so maybe there are some certain things that may happen. So during each and every start of the run, so before each and every start of the run, this system and the software nanopore, sorry, the Oxford Milan, the software will give you an exact idea that how many ports are there still in free condition, which are not basically choked. ধরো আমাদের suppose we have a kind of that pores. So sometimes what happens, যদি কোনো একটা গর্ত থাকে, suppose the pores, মেবি কোনো কোনো গর্ত সেখানে সেখানে চোক হয়ে যেতে পারে, বলি না যে নর্দমাটা ধরো চোক হয়ে গেছে, বা সামথিং দ্যাট হ্যাজ গট
হ্যাঁ,
চোকড, আর বন্ধ হয়ে যেতে পারে। সো এখানেও
হ্যাঁ,
এখানেও সেম থিং দ্যাট মে হ্যাপেন, লাইক what happens basically, many of the ports that initially you have got, so maybe initially you found that up to 4 5 sample run, you found that over 900 ports, 900 ports are free. So maybe as for onwards 6 7 or 10 runs, that you found that at least 100 to 200 ports are already been choked. So maybe 700 ports are still available for your sample run. Now there is another problem is that, as I said, এই সিস্টেমটাকে মিউট করো।
স্যার আপনি মিউট অল করে দিন স্যার।
International Institute of Hotel.
Okay,
স্যার,
yes,
স্যার আপনি
স্যার,
মিউট অল করে দিন স্যার।
স্যার,
yes, yes, বলো।
স্যার বলছিলাম যে
Yes, বলো।
হ্যাঁ, ওই কোয়েশ্চেনটার আনসারটা কি হলো তাহলে, যে এটা যে কেন রিপ্লেস করছে
হ্যাঁ,
না?
আমি কোশ্চেন দিলাম, তাহলে ফার্স্ট কোশ্চেনের আনসার কি দিলাম, যে ইটস নট দ্যাট অলওয়েজ ফর আপ টু 50 রান, বিকজ অ্যাজ দিস ইজ আ ভেরি নিউ টেকনিক, so this has been introduced only in 2012. So this is
আচ্ছা,
constantly evolving system. So the basic system that has been introduced 2012, so that has been substantially improved in 2019, I guess. So as this is
বেশ,
constantly improving from the manufacturer, okay. So but one of the major disadvantages is that in not all 50 sample run you will be getting all 960 ports are available. So sometimes
ওকে,
what happens, as the manufacturer says that 960 ports are available, maybe once you started run a sample of the 30th time, maybe you found that all of the ports have already been choked in spite of adequate cleaning. So if only 100 or 150 ports are still available, but in that case, your sample run will not be completed in a proper way. So in that case, you have to replace your that flow cell. You have to replace your flow cell. So
ওকে,
though it says that, so this is a kind of a 50 sample preparation, but as it is a biological sample, it's not that mathematics that 1,000 years, 100 years back, 2 plus 2 was 4. Today 2 plus 2 again 4, and next after 100 years 2 plus 2 again will remain 4. So as it is not that, so whatever they have proposed, that 50 sample preparation, so 960 nanopores. So maybe it happens that not all cases, even from my experience, as my lab had this facility, so even from my experience, I can share you that our lab was basically focused on cyanobacteria. Okay, so we had at least 5 to 6 different species of cyanobacteria. Some of them are from sweet water cyanobacteria. Some of are from the marine cyanobacteria. But believe me, from my own experience, whatever the thing that we have started running in the nanopore that we have procured, so we could run only 20 to 25 samples per flow cell, and after 20 to 25 run, the flow cell basically got choked completely. So what happens basically, so we had to reopen another fresh flow cell for further run. But one of the advantages that what Oxford nanopore basically gives us, that they basically recycle the thing. Okay, so one thing is that if your flow cell is basically choked and you could not basically run at least a 30 sample, okay, so they basically recycle it. But how to recycle? It's not that it's not a kind of that goods guarantee that okay, so you purchase a TV, so TV is basically got malfunction, now company will replace it. So they will again, or they will only replace once they will go for a rigorous quality checking that in between each and sample that you have run, you actually performed adequate cleaning using their cleaning system. So there are some dedicated software or kind of the thing in their laboratory in United States, so they can easily check that whether in your laboratory, while you basically perform the adequate cleaning in between the sample run. Okay, so once they find that okay, so in spite of adequate cleaning you could not run 50 sample or maybe over 30 sample, so they will basically replace. And replace means, so next time onwards, they will charge. So for one replacement of one flow cell, they will charge only 50% of the cost. They will charge only 50% of the total cost. So
Oh,
again, the 50% is definitely much more cheaper than your pack bio system or your Illumina system. But
স্যার
problem, yes. So
স্যার অ্যাকুরেসি কত কি
so
আছে প্যাক বায়োর অ্যাকুরেসি?
I'm coming. So I haven't basically answered all the queries from your end. So that is
ওকে
the
স্যার।
first thing. So that's why the first thing is that that's why this system cannot completely replace the pack by or Illumina. Now my second point is that in terms of the accuracy, so as I said that in during the base calling is basically there is a rigorous statistical parameter that has been followed. But what happens, the accuracy is not, it's not till date what the manufacturer says that they only achieved around 80% of accuracy. They only
oh,
achieved only
তাহলে তো স্যার প্যাক বায়ো ভালো আরো
হ্যাঁ,
অ্যাকুরেসি বেশি।
no pack bio bhalo, but thing is that if you don't have
কষ্ট।
10 lakh, if you don't have 10 lakhs in your
হ্যাঁ
grant
ওইটা
budget, so
হিউজ হিউজ
if you
অ্যামাউন্ট অফ
don't have,
কস্ট।
so huge amount of cost. Now all the labs in even West Bengal, not all labs can afford 10 lakhs for only sequencing, because
সেটাই
তুমি শুধু সিকোয়েন্স করে কি করবে, তুমি কি খাবে? So
সেটাই।
only sequencing করে তো you can't do nothing, because suppose you have only 900 rupees. Okay, only you have 900 rupees in your pocket, so and you purchased one LPG cylinder with 900 rupees. So then what you do? So do you really consume the gas? No. For that you have to purchase one oven, you have to purchase some cooking material, you have to purchase raw vegetables. Only thing that you can cook for yourself and you can have some food. So that's why thing is that, so that's why for bacteria, so one of the major drawback is that, so what they said that they basically recommended the application of this only in case of still bacteria and in case of the fungi. So not for higher eukaryotic genes. Basically, this Oxford nanopore is still recommended, because you know what?
Oh,
Because you know what? This have only started just 10 years, 2012, not even 10 years completed, isn't it?
oh,
So it is still evolving. And thing is that
yes.
now 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 in each and every case there are some few advantages, there are some few disadvantages. So advantage is that, so from 2012 to 2020, so in 8 years they have dropped down the sequencing total cost in a complete package into 1.5 lakh, which is something really interesting. So at least if you go for some bacterial sequencing, if your lab is only focusing on bacteria, and if you are not rigorously doing the sequencing, maybe you have focused on one or two sample, maybe you are doing some research on one or two bacteria, you can go for at least 10. And if you find that okay, to could conclude your entire research, maybe 10 or 12 continuous sequencing will give you your conclusive result. So maybe this 1.5 lakh or 2 lakhs rupees spending is definitely worthy for you. But maybe if you are really interested in some metagenomic, like you are you are continuously sequencing, maybe 500 sample, 100 sample, 500 sample, 600 sample. So and if you are really doing something really in the mood of whole genome sequencing and de novo sequencing, sort of like that, then I think you must stick to your PacBio platform, because PacBio have around at least over 99% accuracy. And if it is not 99%, so at least over 95% accuracy. And another important thing is that though PacBio and Oxford Nanopore, it gives you a long read sequencing, but there is an bar for Oxford Nanopore that maximum 15 to 20 kb, maximum 15 to 20
Yes, sir.
kb. Whereas in case of PacBio, this is that can be achieved more than 100 kb of a long read. So maybe you can get more than 100 kb of long reads. So that's why as I said, that there are some certain advantages, there are some certain disadvantages. So just I can give you I can give you I can just roll this thing. So why this has happened? Okay, so
Okay.
just okay, so what I basically tried to mention. um,
স্যার এই ন্যানোপোর কেমিস্ট্রিটা একটু বলে দেবেন, আমি
হ্যাঁ
তখন
সব
মানে
বলছি,
কানেকশন লস্ট হয়ে গেছিল।
সব বলছি আমি, জাস্ট ওয়েট। ওকে, ওহো। So this is basically the entire chemistry. Okay, so this is basically the entire chemistry. What happened? So why this is not basically working? Okay. So first thing is that, so this is your DNA. So what is kind of the sample preparation? What is basically the sample preparation? So suppose this is the high molecular weight, the DNA. So just so in two ways that can be done, with fragmentation or without fragmentation. So in both way. So another advantage of the nanopore thing is that, so so in both ways you can perform your sequencing either in a fragmentation or without fragmentation. So that's why it says that optional. So fragmentation is basically optional. So during the long reads, so though the main motto, so main advantage of the Oxford Nanopore was basically a long read sequencing. So you basically try to perform a long read. So thing is that if you don't find any good result, okay, so maybe you can go for an fragmentation also. Now, if you go for an fragmentation, so there is another step for adapter ligation. So there is one condition called adapter ligation. So adapter ligation, and as well as a tether attachment. So thing is that optional thing is that the shearing into the fragments. So first, second thing is that the end preparation. That means you have to prepare the end where you basically go and ligate your adapter. So second thing is that you have to go for some buffer exchange, so that your sample should not contain any salt. And second thing is, and fourth thing is that the library purification. So once the sequencing is done, so how this is basically achieved, so I'm go and basically tell you the detail right now. uh, so the thing is that, so this is basically the area where we basically load our sample. So there is a small groove, can you see here, the small
Yes.
groove is there. So this
Yes.
small groove where we basically put our sample. So after you put the sample, so you find there is a kind of path is there. So a kind of like a এই যে এরকম ভাবে যে path ছুটে যাচ্ছে. So basically, so once this basically done, so here there is a flow cell. So your sample will go and move, and finally one it reaches to the so there is a small part from where your sample will go and reach to the flow cell. And flow cell basically contains your that different. So where is that? So I can I show you this thing? Okay, so one thing, just oh, So I need to show you the flow cell design. Um, okay, so I can right now I think I could not incorporate that flow cell design. So but after you connect your sample with your PC with your workstation with the USB connection, so you will be seeing first screen that will appear on your computer screen, that is this kind of screen, where you can get there are some green area, there are some light green area, there are some deep green area, there are some sky blue area, there are some ash color area. So this green one, so the light green one, so this light green section, so light green section means that how many number of ports are available free through which your DNA sample is basically moving and going, or basically migrating or incorporating. Okay, so these greens are basically the thing is that